individual organism that are identical with genes that are found in modern Homo sapiens, in us, because it would be a stronger evidence, the fact that it kind of looks like a vertebrate and maybe it's in the Cambrian era, but this would be pretty powerful stuff. So they did the analysis, and lo and behold, there were 300 genes, sets of genes, in this creature that are identical to those same genes that are found in us. So those genes have a life minimally of 540 million years. That's impressive. So as I was reading this, I was reminded of a book and a man that I adored the book, and I got to meet the man, and he has recently passed away, named Stephen Jay Gould. Now, Stephen lived in New York and was a guy who was passionate about evolutionary biology, origins of man, origins of life. He was a very ambitious, creative individual. And Stephen wrote a book which was published in 1989 called Wonderful Life. And any of you who love to read, and because we're in a library, you might like to look at Stephen Jay Gould's book called Wonderful Life. And in that book, Stephen talks about something called the Burgess Shale. Now, the Burgess Shale, for some of us, heads are nodding, and for some of us, we've never heard of it before. Burgess was the name of a man who discovered this sediment in the Canadian Rockies north of Vancouver. And in this area, the discovery was made in 1910. And in 1910, they came upon this area, and there's sediment, like hard rock at this point, that is black in color, very, very dark. So everything you see in it is essentially black or black-like. And in it is the impression of the bodies of creatures that lived at the time of when this rock was formed, which ironically is also the Cambrian period. So 540 million years ago, in somewhere in China and somewhere in the Canadian Rockies, and obviously it could be in other parts of the world as well, there was a burst of evolution. Stephen Jay Gould uses the term punctuated evolution. Punctuated meaning exaggerated. So in this window of time, which of course is millions of years in duration, but when you look at it and everything is measured in billions of years, what's a few million years here or there? It's not that big a deal. So when you look at this, they have found already the impressions in the sediment of 65,000 little vertebrates, that most of which are no longer here. So the interpretation is, when there is a burst of evolution, all creatures are not successful. I mean, this is not rocket science. Yeah, I mean, but you would expect that the percentage that were successful would be very high. This is the flip side. Most of these lived long enough to have an impression in the sediment. Now, again, the easiest ones, if you have a shell on your back or tummy, or you have a skeleton, that impression is going to be a little bit different than if you're soft-bodied. And if you're soft-bodied, the impression is maybe a little bit difficult to read. So with microscopes and all of these things, they've documented to date 65,000 different vertebrate attempting to evolve. All of them didn't make it. Those that did make it are our predecessors. And within that crowd was one that's the size of a grain of sand that essentially went on. So as we were talking in the car, I started this just happened Sunday. So as I was reading this and thinking about it and so forth, which I found just fascinating to me, I was wondering what makes for a burst. So, I mean, what could have happened that allowed this enormous attempt at creating diversity in creatures, most of which don't make it, but nevertheless something, because they have evidence of creatures before this time and after this time, and it turns out that the rate of evolution was like tenfold more in the Cambrian, and then it got slowed down to what it is today. And before that, it was essentially what it is today, which means essentially every million years there's a slight change. Every million years there's a slight change. So as a scientist, I formed, or as any of us would do, I formed a hypothesis. And my hypothesis is and was maybe there was a change in ambient oxygen. So maybe the oxygen levels that are hovering above what we call our atmosphere, especially lower to the ground, were elevated in some dramatic way. So help me with this hypothesis. What is responsible for making oxygen? Plants. Photosynthesis. Okay. So where did photosynthesis, this is my mind following, where did photosynthesis begin? Okay. Some creature, not a flowering, not a tree, not a plant, not a bougainvillea, but some one-celled creature, an algae or a bacteria of some type, discovered how to do photosynthesis and could take carbon dioxide out of the air and convert it into oxygen back into the air. And so as you reduce CO2 and raise oxygen, that's like a big deal to primitive evolving creatures. So we're going from creatures that learned how to live and reproduce in so-called anaerobic, the near absence of oxygen, to all of a sudden this rather profound change and so forth. So I then jump on the internet and start plugging in and putting in these keywords and so forth. And so far, as of our drive over here, it's holding up. That actually happened, and there is documentation that the number of algae producing and those simple kind of plants that are able to produce oxygen, there was a soar of them, and they're also imprinted in the Burgess shale that all of this information seems to be pouring out from.
Isn't that the red beds too?
Yes, yes.
And that's the oxygen made the iron precipitate.
Yes, exactly. Like if any of you have ever been visiting in, well, no, I was thinking closer in Arizona, Sedona, where they have this, especially towards the end of the day when the sun is going, it sort of just lights up where it's like an iron fest of sorts. That would be a place where some... Yeah, yeah. Yes?
I want to know how they discovered this creature. If it only the gods who claim advance, how did they distinguish it?
Yeah. If you know, we're all Southern California people. An interesting thing to do, if you have the time and energy to do this, is to go to what used to be called the La Brea Tar Pits, and there is now something called the Page Museum. And inside the Page Museum is actually a working lab that you can volunteer in and mess around. And they have sort of like colanders, except the pores are like itsy bitsy, really tiny. And so you take a sample, which has got all kinds of things in it, and you pour it in and you sift. And so depending on what you're looking for, like the small things are going to stay, and the larger things are going to go through, and that's the technique that was used. But it was a team of Chinese scientists who wanted to find something. So part of it is the need to know or the willingness to go into the field, meaning field work, and be able to look for something. For example, there's the story of the discovery of Lucy in Ethiopia. And nobody sort of said to themselves, I know what I'm going to do today. I'm going to go find Lucy. It was, some of this is like serendipity. And to the informed eye, you sort of like, gee, that looks a little strange over there, and let me dig around a little bit, and all of a sudden, here's a skeleton of some, and then you do carbon dating, isotope dating, look at the geology, and all of a sudden come up with, oh my God, this fossil goes back to a very interesting time in the genesis of Homo sapiens, and we're going to now insert Lucy and the data around Lucy as a piece of evolutionary history as we evolved to eventually get to modern human beings. Yeah. So, yes, please.
They found all these things. My question is the DNA in all of these things, similar, or are there big deviations in DNA?
Right. Good question.
It would be really interesting to know what survived and what didn't.
Right. One line of evidence that we've touched on, we are now in the 6th week.
They all even have DNA.
Right. Yes, they did. We are now in the 6th week, and we're sort of pulling together little pieces that we've touched on along the way. One line of evidence to support evolution: all living things contain DNA. All of the DNA is A, C, T, and G. All of the DNA has a genetic code. The genetic code in creatures of 540 million years ago are using the same code that we're using in 2017. Yeah, true. All living things. So the difference turns out to be the ones that we touched on about microbes that are discovered deep in the ocean, living in these unruly high temperatures, and without oxygen, have figured out to generate and play with sulfur and be able to cope with temperatures that we would melt. So that part of it, that adaptive feature, because everything is about adapting, and we all, everybody in this room, because we are hardy and we're here, we have all gone through the life experience to date by doing a lot of adapting. There's a lot of things that change, and it usually is without notice, and the trick is to sort of go through life with bended knees and an attitude about welcoming change instead of saying, forgive me for this cliche, let's make America great again. It's a pretty great place now. And so anyway, but this is not a political discussion. In this spirit, we've been talking about evolution, and we've been talking about learning, and we've been talking about your futures as learners. So if you could take one of these and pass them, this is the online information and databases to assist you in discovering risk, diagnosis, and candidate gene identification for diseases and disorders related to genomics. There are many of them. I've listed all the ones that are free under the heading free access. And then those, I'm sounding a little bit like the press secretary for, any of you see the Saturday Night Live? Yeah, which was really funny. And anyway, I added two of them, which are you can get to by subscription, in case you are interested in that. But it's just, this is like the need to know, and then this would guide you as to where might you go to get that kind of information. Then also wanted to share with you, this is a paper that will be published next month by yours truly, and it is a special report, and it's called Embracing Precision Dentistry and Medicine. It is written more to clinicians, but you are voyeurs. And so now is the time to prepare for a major transformation in clinical approaches to personalized healthcare. So please, thank you, wonderful. And so the idea is that the things that we're talking about have been very new and evolving in their own sense. And we live in a country which is going through some very complicated issues around healthcare. Because when you set the bar and say all people, that's a big term, all people will have access to quality care. And who pays for it will either be public money or private money, but all people will have access. Once you say that, then it gets hard. Where do you find the money? What does healthcare mean in the 21st century? For everyone in this room, we are people who probably, in our collective, err on the side of preventive services. We want to have a physical exam once a year, or maybe even more often. We want to pay attention to what we eat. We want to pay attention to our numbers. How much do we weigh? Our body mass index, our BMI. We want to walk. Some of us are learning how to do Tai Chi. So we do not impound our knees and so forth as a more, in some sense, a more civilized form of exercise for mature adults than running around the street or doing a very intensive kind of exercise. So what this article is doing is trying to relate. And one of the things in writing it that I thought was really terrific on my part, part of a senior situation here. When you're talking to people about a subject that they haven't personally experienced, using an example of a clinical case can be helpful. And here, Ms. T is a 42-year-old woman who's had a persistent cough for 4 weeks. Could be any of us. During her semi-annual dental hygiene appointment to have her teeth cleaned, the dental hygienist suggests that she consult a physician, and then she gets a chest x-ray and a computed tomography image of her chest, and they discover that she has non-small cell lung cancer. And this was confirmed by doing it would have to be a little invasive biopsy of the lung tissue area where this was done. So if this was done before 2004, the cancer specialist, the oncologist, would offer a treatment in which 10% of the patients would have a positive response. And 90% of the patients, it would be a death diagnosis. Like pancreatic cancer today is for a lot of folks. If she were diagnosed in 2011 or later, like our era, her biopsy would be analyzed for a panel of genetic variants that would predict which therapies might best respond for this particular patient. The goal is to align the specific mutation that causes the cancer with a therapy that specifically targets that and nothing else. So instead of using a sort of generic chemotherapy, which is like a hand grenade thrown into a human body, this would be more like a precise bow and arrow where the arrow is just hitting the respective activity. and in her case, she went into remission, and that activity is called pharmacogenomics. And today, in cancer therapy, the latest, hottest, coolest thing is to use the immune system of ourselves along with a little assistance from the oncologist to attack the cancer. So chemoimmunotherapy is currently best practice for many of these kinds of situations. And then I go on and so forth. But it helps people, whether you're a practicing physician, dentist, nurse, pharmacist, whatever, but somehow putting it in the context of a real life case can be very, very useful. So looking at that you now have in front of you that So the National Institutes of Health, in my opinion, would be called Mecca. This is the largest, most sophisticated, best funded, not-for-profit piece of the American government that we have, and the world envies us. And we currently spend, as taxpayers, $32 billion a year. And then that agency is divided into 21 institutes. And each of those institutes, I've listed a group of them, and they are like the Cancer Institute, the Heart Institute, the Communicative Disorder Institute, the Digestion and Kidney Institute, and so forth. And then there's one called clinical trials. You're told that something's bad with you and it's rare and unusual, and this would be an invaluable place to go for you or a loved one to look at, are there any clinical trials in the country, and what are they about, and what are they attempting to show, and then be able to go to your primary care physician and ask him or her to be your advocate. Could you become part of that clinical trial, whatever? So it could be pancreatic cancer, it could be small lung cancer, it could be anything. But that is very, very useful.
What's the difference between small cell lung cancer and large cell lung cancer?
Well, I mean, not being glib, it's the size of the cell. The cells in the lung that this is looking at are called type 2 epithelial cells that are involved in respiration, and they alter their size. In this malignancy that she was diagnosed, they round up and become spherical, and in other patients they become elongated, and therefore the term small as opposed to the other thing is there's one here called phenomizer. It's about halfway down the page, and that is the attempt of linking genotype and phenotype and being able to drive. That's something that we've been discussing over the last 5 weeks along the way. The next one down, Entree, is a query for being able to interpret any genetic information that comes along your way. Then last week we talked about stem cells and the California Institute Stem Cell Research Initiative, which was born in 2004 by the California voters, putting aside $3 billion, and that is going strong. And that is something where you can look at the progress made in, for example, spinal cord injuries. There was a time where the spinal cord injury essentially was you shrug, like there's nothing we can do. And as time has gone by putting in stem cells, they've been able to have a number of patients go into remission. And so this means taking a stem cell. Stem cells come in 2 flavors, as we touched on briefly. One flavor are called embryonic stem cells. And this sort of kicked off a very active political engagement between religious beliefs and might be called less religious beliefs, and that we should not be using human embryos for experimentation and so forth. So a moratorium was placed on anyone who would receive federal money could not do embryonic stem cells derived from humans. If you were a private company in the United States. You can do what you want to do. These rules only apply to federally sponsored research. So if you're a company called Amgen out in Thousand Oaks, you could continue to mess around with human stem cells, but if you were UCLA or USC or any of the universities that are connected with federally grant activities, there was a moratorium. Other countries and other parts of the world have different rules. The United Kingdom never stopped working on human embryonic stem cells, and so they continued, and some of the pioneering work came out of England for that reason. So nowadays, ironically, in South Korea, a group there made a remarkable breakthrough. They took skin, a biopsy of skin from an adult. They then teased off the outer covering, the so-called epidermis, from the dermis. And in the dermis, there are lots of cells that are stem cells. They would be called adult mesenchymal stem cells. And then they asked, if we put in 4 regulatory proteins that work to regulate gene expression, could we convert the mesenchymal adult stem cell to become embryonic? They succeeded. They formed a company. They wrote a patent, all of those things that go on. And so that became the new state of the art. There was no need to have an argument. There was no need to posture over when does life begin and all of those kind of questions, which become very difficult to sort through a priori. And so today, the adult stem cells can be used to create literally embryos. I mean, it is phenomenal. Yes. It's a South Korean. I don't recall the. You can look it up, but I don't recall the name. And an adult stem cell company that works on making new bone, new muscle, new cartilage is called Osiris, and it's in Baltimore. That's one I remember, but I don't remember that one. So picture this. I'm fairly trim, probably could always lose a couple of pounds. But where do you think in my body, if I needed adult stem cells, would be the very best place to get them?
Your skin.
Okay, so it could be skin. Okay. Fat. It turns out that fat has the highest concentration of stem cells of any tissue in the body. So the technique called liposuction, where you go in. Now, before this discovery was made, they would go in to do liposuction on a man or a woman and throw the fat away because it was like to be discarded. No more. Now, you extract the stem cells, clean them up, wash off the lipids and so forth, and then repurpose them by either modifying them with these 4, they're called transcription factors, and then they can become the equivalent of embryonic stem cells, or you can use them for an application like I broke a bone and I heal slowly, and so could you give me a hand using my own mesenchymal stem cells, which means no immune response. So I have a sister named Leslie. If I take a graft from my sister, I have to be put on immunosuppressive drugs because as much as I love my sister Leslie, my immune system recognizes her as foreign and is going to do everything it can to reject those cells or tissue or whatever that came from her. On the other hand, if they can come from ourselves and then just be amplified, and almost everybody's got a tummy or small or large that could be utilized in this way. It's a very cool thing. So that's one to look at. And pardon me? Are they doing this routinely at any of our medical? They're doing it routinely at SC. They're doing routinely at Stanford, UCLA. It's one of the tricks. When you are an orthopedic surgeon type, and you're looking at a patient where there is a need to augment bone, you evaluate the patient. It could be as simple as I just put these two together, and that's going to be the least expensive, fastest, most predictable outcome. On the other hand, you, and especially because you're a wild woman, you drive a motorcycle, and you've got a compound fracture in which some of the bone is missing. So it's not as simple as two broken ends back together again, but it's like stuff missing. In that case, an artificial tube that would be biodegradable filled with mesenchymal stem cells would be offered to you as one way to go. Another would be to take bone from your hip, move it into the area, again, depending on size, depending on age, depending on, all of the things that are part of our uniqueness as when we become patients, it would be interpreted that way. But it's one of the tools that today exist in the hands of orthopedic surgeons for bone healing, cartilage healing. They're used in knee replacements. They're used in hip replacements as, one more approach. Someday, right now we use titanium or titanium-like materials in hip replacements or even in knee replacements, and the idea would be to get away from an artificial material altogether and be able to use a so-called a biological solution to attain the result that you want. And we've shown that picture a couple times of somebody with nasal cell carcinoma lost their nose. The nose is essentially not made out of bone, but it's made out of cartilage. Cartilage is one of the few tissues in the body that loves blood to be diffused to it, but doesn't have blood vessels in it. It's called avascular, which means, in a surgical sense, could you make cartilage to replace the cartilage that was taken away because of nasal cell carcinoma, put a skin graft on top of it, and the person walks away with a perfectly attractive nose. And I showed the work that was done originally in Boston. That's what they did. You could do the same thing with the ear. Again, the ear is cartilage wrapped with skin as opposed to having a dependency on blood supply. You can do the same thing with ears that you can with nose. Yes? Yes. So in 1997, the FDA ruled in favor of allowing a company, I don't remember the name of the company, to be able to do knee repairs using mesenchymal stem cells. All of this started in 1967 with childhood leukemia. So kids are born, and some of them, they discover that they have leukemia, and it's referred to as childhood leukemia because the onset is like 2 years of age, 3 years of age, very young. So and it was an NIH discovery that if you do a bone marrow transplant, so bone marrow, maybe everybody here has been in a butcher shop and had a view of bone marrow at one point or another. Bone marrow is sort of like red, pink color running down the shaft of a big bone from a cow or something like that. Well, that material, bone marrow, it is loaded with stem cells to make white blood cells or red blood cells or mesenchymal stem cells. So there are three kinds of stem cells that pack into the bone marrow of our skeleton for all of us. Now, why that becomes, it's sort of a clever evolutionary strategy. Bone marrow is like it's everywhere. It's in our fingers. It's up, going up. Wherever our skeleton goes, there's a little bone marrow. Well, every 120 days, the red blood cells that I made today are going to be dead. I must replace my red blood cells every 120 days. Well, for some of us, that's a short time. For some of us, it might be considered a long time, but it's 120 days. So that means my bone marrow needs to come up with the equivalent number every 120 days. And Pablo and I like to volunteer and give our blood. We're both type O donors. We're so-called universal donors. Well, that means when I give a couple of pints of blood, I got to replace that, or it's the end of giving blood, and plus I would start getting sleepy and tired, and it would be like suffering from anemia, where I'm just not able to carry oxygen, because the red blood cells have a molecule called hemoglobin, who has the job of carrying oxygen around to the parts of my body. My brain, my skin, my whatever part comes to mind needs oxygen. We are aerobic dependent. We need photosynthesis to keep cranking out oxygen on this. And as our population on Earth went from, a couple of us are reading a book called Sapiens, when we went from hundreds of people on Earth to thousands to millions, and by 1900 the Earth had 1 billion people. Well, in 2017 we are now 8 or 9 billion people. 8 or 9-fold increase in 100 years. This is a stress on oxygen, on water supply, on food supply, on housing, traffic. In Los Angeles, you could believe it easily, but other areas a little bit less so. Anyway, then dropping down, one of these is called the National Academies Press. So this is a distinguished group. It's the National Academy of Engineering and the National Academy of Sciences and the National Academy of Medicine. They're all located in Washington, D.C. on Constitution Avenue in a beautiful building called the National Academy of Sciences. There's a fantastic statue of Albert Einstein in the courtyard that's fun to sit in his lap. It's huge, and have your picture taken if you ever travel to D.C. But anyway, they publish reports that are either called reports or workshops or summary on a regular basis, and they are free. So if you go to that site and just, depending on your own interests, look at what is being offered. Certain times of the year they come out, for example, with these wonderful books about women becoming scientists, and therefore, like for adolescent young women, to inspire them to consider staying in school, doing math, and all of that kind of stuff. Wonderful activity. Then we've been talking about CRISPR and the ability to do gene editing, and I thought you should have access to what I was drawing on. It's called the International Summit Human Gene Editing, a global discussion, and then this is the website, and you can either read it from the screen of your computer,